This is the Physical Activity Researcher Podcast, a podcast for researchers of sedentary behavior, physical activity, and sports. Join for a relaxed dialogue about research design, practicalities, and, well, anything related to research. Learn from your fellow researchers useful and relevant information that does not fit into formal content and limited space of scientific publications. And here is your host, researcher and entrepreneur, Olli Tikkanen.
Welcome, everyone. I'm very excited about today's guest. His main research interest is muscle metabolism in diabetes and obesity. He has done his PhD in the University of Aberdeen in Scotland. Currently, he works as a postdoctoral researcher in Karolinska Institute in Sweden, and he is a visiting fellow at Edinburgh University. Ladies and gentlemen, please welcome Dr. Brendan Gabriel. Welcome, Brendan.
Hi, Ollie. Thanks for having me.
Yeah, no, great to have you. So, what kind of research you are doing at the moment?
So, for the last roughly 4 years, mainly in Karolinska, I've also been visiting Copenhagen University in a collaborative project. I've been looking at how circadian rhythms, particularly the molecular circadian rhythms, might be disrupted in the peripheral tissues of people with metabolic diseases such as diabetes. And we're particularly interested in skeletal muscle. And, that's quite an important tissue in terms of metabolism and also in terms of the ability to exercise, which, as I'm sure the listeners are aware, has a lot of health benefits for those type of diseases. So that's what I'm mainly doing at the moment. So it's quite interesting.
Yeah, and so I have understood that the 2017 Nobel Prize was awarded for the finding of biological clock.
Yes,
and right. Yeah, and then I read something about the period, timeless, and double time genes. Sure. Could you explain more about those genes and how does this biological clock actually works?
Of course. So I think most of the listeners will probably be familiar with, when you talk about circadian rhythms in biology, you think of some of the kind of obvious things that we can observe. So things like the sleep-wake cycle, body temperature, heart rate, things like that. And there's also things the listeners might know of in terms of hormonal circadian rhythms, such as melatonin, which is involved with the sleep-wake cycle and other hormones, such as cortisol. What people who aren't molecular biologists might not know is that there's also, within every cell in our body, there's a group of molecules which actually act effectively as a clock within the cell. And that was what really the discovery of these genes and how they work was what the Nobel Prize was awarded for. That was in 2017, awarded here at or from on the behalf of the Karolinska Institute. And these molecules, they are produced from genes, which then produce proteins, and these proteins then activate other genes which inhibit their own production. So that's what we call an autoregulatory feedback loop. And this control loop sort of self-regulates over roughly a 24-hour period. And I find that incredible, and it's very well conserved throughout evolution. So we, biologists believe that these rhythms may have originally evolved probably millions of years ago as a response to fluctuations in UV light throughout the day. So UV light can be damaging if you don't have the correct sort of protective biology in place. So there might have been these genes that acted in order to produce certain molecules which protected the cell from UV light. But now, in modern biology, obviously that's not as much of an issue as we become more complex organisms. But what is an issue is our response to the diurnal nature of the world we live in. And that's, of course, in terms of sleep-wake cycles, in terms of our activity, which, obviously we're walking about, moving about throughout the day. And of course, we're resting at night. and also in terms of what we eat. So obviously we do the majority of our eating, during the daylight hours or sometimes into the early evening, and we what we see particularly in these metabolic diseases is that the body requires certain metabolic processes to be primed and ready for these kind of metabolic events, and that might not work so well in certain metabolic diseases. So it's really interesting, particularly in muscle. We think actually almost 2/3 of the transcriptome, so the genes that are active in muscle, may actually come under the control of these core clock genes. So some of the genes, for instance, that you were talking about, such as purr and cry. So you mentioned timeless. That's in Drosophila in flies, which was originally discovered. And then purr and cry and clock and BMAL are the mammalian versions of those clock genes that were originally discovered in Drosophila. But we think that almost 2/3 of the genome might respond to these fluctuations, which the origin of which happens very often in these original clock genes. So I think it's an emerging area of research, and it seems to link to a lot of important metabolic processes, particularly in skeletal muscle. I think that's a kind of summation of it, and it's a fascinating area, I think.
I fully agree. So you said that these genes and these proteins have an effect on a lot of skeletal tissue metabolism. Could you tell what kind of effects you have already found?
Sure. So if you look at work by Schifano and Kenneth Dyer, they've and also Karen Esser, so researchers like that, and many others, particularly in skeletal muscle, they've done this process called chip sequencing. I don't need to fully explain what that is, but essentially it's looking at what these clock genes interact with and what pathways they kind of may control. And a lot of the pathways they find in terms of the metabolic pathways are things like lipid metabolism, glycolysis, so that's breaking down, burning sugars, carbohydrates, and also even protein metabolism, and particularly interestingly, mitochondrial metabolism seems to pop up a lot. So mitochondria are very important in skeletal muscle. They're the sort of powerhouses of the cell. They produce a huge amount of chemical energy that we need, and they also break down a lot of these metabolites, these molecules that our cells are exposed to after a meal. So it's very important, and it seems to be this sort of axis of sort of diurnal metabolism in the muscle, where these clocks within the muscle are sort of turning the dial on how active these metabolic pathways are.
All right, and what kind of practical things you see that it has an effect on fat metabolism, glucose, protein? How big is the effect in the circadian rhythm, and what kind of practical things you think there are?
Yeah, so I think what is hard to say for sure about the practicalities of it is there's a lot of confounding factors in this research when we take things from a molecular level into in vivo research, so into the human body. So we can't discount the sort of behavioral diurnal rhythms that I mentioned earlier, and how, whether it's cause or effect. So I think we have to say that it's a complex interaction, and we can't really discern whether it's cause or effect yet. But it's clear from, so if you look at Patrick Schuin's work, they're a group in the Netherlands. They've done really nice work with human subjects where they can take muscle biopsies, so little pieces of their muscle, and measure the metabolism of those biopsies. Really while they're still really fresh from coming out of the human body, and they can see that the mitochondrial metabolism, the sort of ability of the muscle to oxidize substrates, to burn through sugar and fats, is slightly higher in the evening in muscle, and that sort of relates to what we see, what other researchers have seen and what we see as well, in terms of exercise. That exercise capacity nearly always seems to be higher in the afternoon, evening, almost irregardless of whether you're a morning person or not. Exercise capacity seems to be higher in the afternoon and evening, and that's particularly obvious in high intensity exercise, so more the strength, high intensity interval exercise. One's capacity to perform that seems to be higher in the afternoon and evening. So that's some of the sort of practical realities we observe in the literature from that. And as I said, it's very hard to know whether that's a result of these clock genes, or whether there's an interaction of factors as well. So yeah, that's some of the practicalities that we know about that.
Yeah, and have I understood correctly that these clock genes, they are not influenced by the light, but they work with the 24-hour cycle independent of the light?
That's exactly right. So, really interesting. What we can do is take, so for instance, we take muscle biopsies. We then get some of the sort of stem cells that are present in the muscle, culture them completely independently of the body, and we can grow those cells for a few weeks. And then when we do something to synchronize all the cells so that their clocks are all at the same time, we still observe the complete functionality of this clock pathway, and it oscillates over roughly a 24-hour cycle. So that's in cells in a dark incubator, which have no access to light, or virtually no access to light, and certainly no ability to sense light. We still see these autoregulatory, these self-regulating machinery in the cell, and we can measure the expression of those genes and how much they're turned on or off. And it's almost exactly a 24-hour cycle, which I find amazing.
Yeah. And it's just an idea that came to my mind now is that could you actually test that whether this change in exercise capacity is related to these genes, or actually the light cycle, that if you have people in the dark or modified light cycle inside, and then if you see the same pattern in the exercise performance, then you would probably think that they are made due to these clock genes or clock proteins.
Exactly. So, so often in research, when you're interested in a set of genes, you would sort of manipulate their regulation in a mouse model. Obviously, it's pretty hard to do that in humans. So, for instance, the pair and cry genes, I think there's pretty good evidence in mice that they actually, so these genes are the sort of inhibitory arm of the self-regulating feedback loop. So they tend to be expressed in humans at a higher level in the nighttime, when we should be sort of sleeping and resting. And those genes seem to inhibit exercise capacity. So if you knock them out in mice, the mice actually perform slightly better in exercise. So I think there's definitely evidence in mice that the core clock does control exercise capacity to some extent. Obviously, it's much harder to test that in humans. There's some evidence that mutations in these core clock genes have functional relevance in terms of sleeping ability. So people who have disrupted sleeping patterns are also associated with mutations in some of these genes. So I think there's certainly a functional relevance to these clock genes, and it's not just in the brain, which obviously controls or predominantly controls the sleep-wake cycle. It seems to also be relevant to exercise in the peripheral tissues such as muscle, and in terms of metabolism as well. There's some very initial emergent evidence that mutations in these genes might actually change the way we sort of metabolize or take up sugar in response to insulin. But that's at the initial stages. But I think for exercise, as much as we can say from mouse models, it seems that they definitely do influence exercise capacity, and the opposite is true as well. So, when human subjects exercise, it changes whether these genes are turned on or off. So we can actually sort of contract cells in culture, again outside the body, and use electrical pulse stimulation to actually contract the cells, and that changes the rhythm of genes. So we see this rhythm, then we contract the cells, and then the rhythm is shifted. So we see a phase shift in the rhythm due to exercise, and that's observed in humans as well, and in mice. So we think that's pretty well conserved, that these clock genes are sensing muscle contraction or exercise. So moving forward to sort of disrupted sleeping patterns, potentially to shift work, things like that. That might be a strategy where you can sort of time exercise bouts in order to kind of optimize these molecular circadian rhythms.
So, for example, for athletes, if they need to compete in a different time zone and it's an important competition, do you think they can reset or change the timing of these genes by exercising in different times, maybe nighttime?
Yeah, for sure. I think I think generally when we apply molecular biology to sport, I think it's often results in us telling athletes and coaches something they already know, and I think that's probably true here. I think I think that's pretty well practiced in coaching and sport, that athletes will try and adapt to a time zone change as much as they can. Obviously, I think there's practical implications in that. You obviously have to optimize training as much as you can, and changing times of training can be difficult for a variety of reasons. But yeah, I think there's good evidence that you can mitigate the change in performance at time of day by getting adapted to a different time of day. So and particularly, like you say, going eastwards generally seems to sort of affect people more. So if you're going eastwards around the globe, you can if you can try and start exercising and training at the new time as soon as possible, or even before travel, that seems to sort of mitigate some of the deleterious or negative effects of the shift in circadian rhythm.
Yeah, makes sense.
Let's have a short break and hear a few words from our sponsor.
This podcast is sponsored by Fibion, a research device that has been shown to be valid in tracking sitting, standing, physical activity and energy expenditure. Furthermore, Fibion has been shown to be valid categorizing physical activity into light, moderate and vigorous intensity. In addition to scientific accuracy, Fibion provides automatically produced and easy-to-understand reports for research participants. Get scientific validation and learn more about Fibion at fibion.com/research.
And I noticed you have a new paper in preprint called Transcriptomic Profiling of Skeletal Muscle Adaptations to Exercise and Inactivity. It sounds very interesting paper. Could you tell us more about this study?
Sure. So that study was led by Nicolas Pilon and Julien Zeraf. I contributed a little bit to it. But the idea behind that was often in exercise physiology, I think we end up, because the studies are pretty time intensive and resource intensive, we often end up with underpowered studies. And what I mean by that is they have not enough subjects in order to sort of say for certain that statistical changes are relevant. And that's particularly the case, I think, often with studies which take samples of the muscle at different times and then look at gene expression with these sort of omics techniques. So omics is just anything where you do a lot of you look a lot of molecular phenomena at the same time. So we're looking at sort of transcriptomics, which is how much genes are turned on or off. And that's very important in terms of saying the effect of exercise within the muscle, the effects of different types of exercise, and whether we can sort of, almost reverse engineer the effects of exercise and understand how it actually works in order to get a better understanding of biology. So what Nico did on this was he led a research project that sort of took publicly available studies, so studies that had already been done, that were maybe smaller numbers of people. So maybe a lot of them were, around 10 to 20 subjects. They'd had their muscles sampled in terms of different types of exercise, different types of subjects. So some of them were healthy, some of them were slightly older, some of them were diseased metabolically. And then he sort of looked at the transcriptome of all these studies put together. So that really increases the statistical power, gives you a lot more ability to say for sure, whether a result is actually real. If you see it repeated, for instance, in several similar scenarios, then you can probably say it's real. Alternatively, it also gives you the ability to better distinguish different types of exercise. So we see, there's a lot of similarities between acute exercise, but there's also, pretty distinct pathways, for instance, between resistance training, aerobic training, and then the sort of in the middle kind of training such as HIIT, which seems to have some of the beneficial effects of both types of training. And then that really seems to be reinforced after training. So when you get a chronic period of exercise of about 4 to 12 weeks, you start to see these divergent pathways between aerobic and resistance exercise in particular. And interestingly, we see that, for instance, diabetic subjects, exercise is very good for them and has a lot of beneficial metabolic consequences, but they don't seem to respond quite the same way as healthy subjects that are sort of matched. So that's quite an interesting finding that maybe hasn't been so obvious in the field before, but because we've collated all this data, we can see that a little bit more clearly. So I think that's something that's quite interesting.
Yeah. And how do you think there's always, for example, when you have aerobic training intervention studies, there's always non-responders. Do you think the metabolic pathways could explain the non-responders?
I think it's a very complicated topic in terms of non-responders. And I think, in absolute terms, I don't think you ever get someone who doesn't respond to exercise in some way. It's maybe that you just didn't measure how they responded. And I think, there's a lot of complex traits and environmental factors that contribute to response or non-response. So I think, there's obviously inherited factors that can contribute to that. But there's also, environmental factors and the types of training that people may respond or not to. There's some studies suggesting that people who don't respond to a certain type of exercise, if you give them a different stimulus, then it can work differently. So that might speak to this sort of idea that exercise regimes should become more personalized, that potentially we can sort of predict what kind of exercise certain types of people will be most beneficial for those people. So that could be something really interesting. And I think that could potentially come back to, if people have severely disrupted circadian rhythms, perhaps in the future, we would be at a point of saying, well, if you did an exercise at a certain time of day, that might be more beneficial for your response to exercise. So yeah, I think, a very complicated area, but, there's emerging research in that suggests, potentially we could give more specific exercise recommendations, but, I don't think we're there yet.
And is there any studies that that have looked at like training intervention studies with disrupted circadian rhythm, people with that, or?
Yeah, I don't think there's been many. So there's been a few with shift workers. Generally, they're quite acute studies. So firstly, just to say, there's a few studies that have artificially disrupted people's circadian rhythm. So taking them from being awake during the day and sleeping at night, and they develop, deleterious metabolic effects within a matter of days. So that, including sort of insulin resistance, which is also is involved in the pathology of diabetes, type 2 diabetes. So that can occur within a matter of days. It is on that acute level, it is reversible. So it's not the be all and it's not the be all and end all. It's not completely irreversible. So that's why shift work, or one of the reasons we think shift work is associated with metabolic disease, increased risk of metabolic disease, and other sort of comorbidities. And some research suggests that, it certainly seems that exercise, being fitter, seems to give you a more robust circadian rhythm. So, there's a study, again, from the Netherlands that showed that trained subjects, when you take their muscle cells and grow them, again, independently, so externally from the body, they have these higher amplitudes, these more robust circadian rhythms in their core molecular clock. And in terms of sort of actual practical implications in shift work, there was a study where the subjects did exercise every 15 minutes, every hour of their shift, and that seemed to have beneficial effects. But obviously, that's kind of like a proof of concept. It's not practical in reality for, for instance, nurses to be leaving their patients every hour and doing 15 minutes exercise. But I think that group as well did exercise before the shift, and they saw some beneficial effects of that in terms of body temperature regulation and sleep once the shift was finished. So I think at the moment, we can maybe say that the most promising timing of exercise in terms of shift work might be sort of before the shift, in order to sort of prime the body for alertness, being awake. But again, there's a lot of research still to be done on that before we can give really concrete practical recommendations.
Yeah, and in this transcriptomic profiling study, you were also looking in activity. Did you find anything interesting related to inactivity?
Yeah, so I think inactivity, it seems in general it has a very, it has an almost opposite response to acute activity. So, there's downregulation of sort of mitochondrial pathways. There's sort of decreased ability for the cells to, burn, oxidize substrates such as glucose and fat, which is generally associated with worsened metabolism in terms of disease progression. There are some specific pathways that seem to sort of be specific to inactivity rather than just the opposite of activity. But we're not entirely, I don't think we can entirely say exactly how that works yet, or whether that's a consequence of the study design or how we've looked at it. So there haven't been that many studies looking at inactivity that in a sort of matched and controlled way to compare it to activity. So I think that there needs to be a lot more work done in that field, particularly looking at, for instance, people going in for operations in hospital. They can be virtually immobilized for days, sometimes weeks. And that's, that can be a planned, scheduled operation. And I think more should be done to understand what is happening to those patients metabolically. And if there's anything we can kind of do to mitigate that, I think that would be, that's a really, really important area. A lot of those patients might not be able to do exercise or activity. So we need to understand the flip side and what's happening to, biologically when they're inactive. So I think that's a really important kind of emerging topic. But again, as I said, we don't have the enough properly controlled studies to really do that comparison yet.
I see. And in this study, like what kind of data you had of exercise and inactivity? Were there like acute effects or?
Yeah, so within very, very quickly with inactivity, so within a matter of weeks, if not even days, there's downregulation of these genes are turned off effectively that are important in particularly sort of mitochondrial metabolism, and as I said, the ability to sort of take for the muscle. This is only talking about the muscle, just to be clear, for the muscle to take in sugars, fats, and to burn those sugars and fats. So essentially, not dissimilar to when the body's getting ready for sleep. It's dialing down the sort of pathways that it doesn't see as necessary, and it might sort of have a corresponding increase in more sort of inert kind of structural pathways, but in general, it's dialing down the ability to sort of use fuel to make energy, which obviously is in an evolutionary sense conserves protein. It conserves important kind of molecules in the body, but in terms of the current modern world we live in, where we have constant access to food, that's not necessarily a good thing, and can often be kind of associated with disease pathology, particularly metabolic disease.
Okay, let's get back to that in a moment and hear a few words from our sponsors.
This podcast is sponsored by Fibion, a research device that has been shown to be valid in tracking sitting, standing, physical activity and energy expenditure. Get scientific validation and learn more about Fibion at fibion.com/research.
So basically, there's a lot of studies showing that long periods of inactivity, even just 1 hour, has adverse effects to your health. So you think that is dialing down certain genes because it seems that we don't need the energy for the activity, so the body is preserving those energy stores. Do you think?
Yeah, I think it's again. I think it's one of these things that we can't say that the dialing down of the genes is not necessarily causal. Well, it will eventually be causal, but I think there's also a decrease in a very acute sense in terms of minutes or hours. For instance, people going to an office job and sitting down. There's a immediate kind of physiological effect of decreased blood flow to the muscles. So then, they're not having as much lipid and sugar delivered to them. So they're not able to use that as a substrate. And contraction itself of muscle has this sort of effect of increasing glucose transporter movement to the cell surface. So the ability to take up sugar is sort of automatically increased with muscle contraction. So I think that's very, very important. And there have been these studies looking at sitting time, sedentary time associated with disease. I think, in my personal opinion, I think a lot of that is simply the inverse of activity. I think the more exercise someone can do, generally the healthier they are, as long as that's done in a healthy, controlled setting. And I think obviously sitting down, not doing exercise is unhealthy, mainly because you're not doing exercise. I think there might be some distinct divergent kind of innate qualities of inactivity. As I said, I don't think we've done enough controlled match studies to really kind of tease out what they exactly are yet.
Now that's really interesting. So basically, I think it comes down also to threshold. You were saying that it's kind of not having exercise, but in exercise you basically have like quite considerably higher energy expenditure. But if you, for example, compare sitting and standing, the energy expenditure difference is really small, mainly depending on your body weight. or how active you are while standing. But basically with the muscle contraction, there's quite a bit of difference, because you need to activate muscles even when you're standing still, that you kind of oppose the gravity. So do you think that it's from the pathways, it can be that even really small muscle activation can be enough for these
Yeah,
genes as a stimulus? Yeah.
I think in the sitting standing, firstly, I'll just say, yeah, like you said, that it doesn't seem to be, in terms of metabolism, there doesn't seem to be that many benefits to standing. I think it might be something we haven't really elucidated yet, such as sort of blood flow changes. And for instance, I personally pretty much spend the whole day standing, and it's not for metabolic health reasons. It's because I have a sore back when I sit down. So I think there's other, health reasons why standing is probably, well, it's probably good to change between sitting and standing for sure. I think when you, when you work in an office all day. But as you say, I don't think getting everyone to stand at an office is going to solve the obesity crisis, for instance. And I think you're entirely right that the turnover of ATP increases enormously in muscle during exercise, and again, like you say, it's a threshold. So, and what sort of any exercise, over 60% VO2 max, 50 to 60% VO2 max, the turnover of ATP, increases enormously. And of course, we need to make that ATP from somewhere, and that's taking in fuel, taking in substrates, and making ATP from that. And that is, one of the muscle is absolutely designed to do that and turn it into chemical energy, which enables muscle contraction. Muscle is a very sort of focused kind of tissue in that regard. So, and the body is really clever in the way that it then, the consequences of that metabolic changes, so that the increase in substrate breakdown, increase in ATP production, produces metabolites and molecules, which then signal to the DNA, to the sort of the transcriptional machinery that turns on and off genes, which enables the muscle, the cell, to make the proteins, the RNA, whatever else it needs in order to be better at that job next time. So it's better at kind of breaking down substrates, better at making ATP, better at contraction. So, that's the idea with repeated bouts of training. You get this acute effect in the muscle, this acute metabolic effect, which has an acute effect on genes. Then over a period of time, that repeated effect turns into a chronic effect, which has a slightly different signature in terms of the proteome, in terms of the transcriptome, in terms of the metabolome, in different scenarios to acute exercise. And that's what we were really trying to elucidate in that paper with the transcriptomics of all these collated studies.
I think it's really, really interesting way of studying. And I'm still thinking, like, for example, in bone remodeling, it has been found that you don't need many strong accelerations or kind of impact for the bone, that just few per day is enough for the bone to remodel. I'm thinking, could it be also for the muscle that maybe if you do like 2 seconds per day, like maximum contraction, could it be that it affects the pathway already, or what do you think?
I think, so yeah, I guess like looking at this kind of sprint training and trying to do that, I think we don't really have the evidence on the long term to know how that affects health. For sure, doing very intense exercise, I think it is, as long as it's sufficient to generally to reach exhaustion at some point, it does seem to have an effect on the transcriptome. I think, the bigger and more extensive the exercise bout is, generally that has more of an effect. So, a lot of this is in relation to this often cited reason that, a lot of people say when they're asking the questionnaires, that the reason they don't do more exercise is time. I'm not, sometimes I'm not sure whether I entirely believe that. I think the relationship between what people say on questionnaires and real environmental reasons doesn't always completely tally up. I was asked on another podcast what I thought we could do on a societal level to kind of get people to do more exercise and be healthier. And again, as a caveat, this isn't my area at all, but on my personal opinion is that facilitating cities so that people can do exercise more easily and build it into their daily routine. So, for instance, commuting, being able to walk or cycle, or even run or kayak as you can in Stockholm to work, I think is a fantastic way of increasing people's ability to do exercise, and that's of course going to tie into this transcriptomic effect that we see, because again, the more intense the exercise bout, the longer the exercise bout, the more often the exercise bout is done within reason. You obviously need periods of recovery. That has a more robust effect on the chronic, in terms of the chronic kind of transcriptomic, metabolomic, proteomic regulation. So, of course, this, as I said earlier, this all feeds back into, us molecular biologists telling coaches and athletes and people who are researching physical activity, what they already know, that, the more exercise you do, generally, the more effect it has and the better it is. Of course, as I said, you need periods of recovery in that as well. But, that's generally the case, I think.
Yeah. And also you have another paper which is about exercise timing and type 2 diabetes. Could you tell more about this study?
Sure. So what we did there was, we were doing a lot of this molecular work, and we see potentially that the molecular clock in diabetic muscle is kind of dysregulated. It seems to have a smaller amplitude, seems to have less genes that met our statistical criteria for being called circadian genes. And we wanted to kind of see, in a parallel study, whether that has any sort of consequences in the real world. So, what we did, we just got 11 type 2 diabetics from Stockholm. We fitted them with continuous glucose monitors, which is obviously a really good way to measure glycemia levels during the 24-hour cycle. And we had these monitors on for, I think it was 4 weeks in total. So what we took a baseline measurement first for 2 weeks, just when the guys were going about their normal day lives. And then we got them to do sort of controlled high intensity interval exercise for 2 weeks, every other day, either in the morning or in the afternoon. We split the group randomly into half, and then we gave the subjects a 2-week period just to wash out any of the effects, and then they did the opposite. So if they've been training in the afternoon, they then switch to the morning, and vice versa. Not generally in terms of glycemia, we generally expect, as I was saying, muscle contraction tends to move these glucose transporters. It tends to increase blood flow to the muscle. It tends to have the effect of acutely reducing blood sugar, glycemia, because the muscle takes up more sugar in order to use, again, to break down and to make ATP for contraction, and also to store. There's another effect I won't go into, but it also wants to store more glucose for the next time that is challenged, basically. So in the afternoon exercise, we saw that when the subjects did exercise, afterwards and a little bit the day after as well, compared to the baseline readings we took, which we compared, day to day. If it was a Wednesday, we compared to a Wednesday. We saw that indeed exercise did reduce glycemia. But surprisingly, when the subjects exercised in the morning, actually glycemia slightly increased compared to basal. So it increased compared to the afternoon exercise, but it also increased compared to basal. Now, this was a study where we didn't control a lot. It was kind of a pilot study. We wanted to see the effects of what would happen in the real world, outside of the lab. And what happens when you just give these guys the exercise to do and don't control the rest of their lives. So we're not really sure on the reason for that, but it might be an adaptive thing. So towards the end of the 2 weeks, we saw that that effect was less severe. There was less increase in glycemia. So it might be that they're just not used to working that hard, doing intense exercise in the morning. After a few weeks, once they become acclimated to that, they might have a normal kind of exercise effect. It might also be changing their sleep-wake cycle potentially. So we saw actually there was a sort of anticipatory increase in glycemia. So even in the early morning when these subjects were probably sleeping or only just waking up, there was this anticipatory kind of increase in glycemia. So as I said, we're not entirely sure how that works, and I certainly don't think that we can conclude from that the morning exercise is bad for diabetics. I think there's work from other groups. So if you look at Javier Gonzalez, he's been looking at healthy subjects and people with obesity as well, and he's found some beneficial effects of exercising before breakfast, and not necessarily looking so much at timing, but before or after breakfast. That's another one of these kind of physiological effects that can change the molecular clock. So obviously eating foods can really change the way our metabolic processes are working, and exercising before or after that kind of profound metabolic stimuli can have these changes in metabolism. And actually his work suggests that there might be some beneficial effects in terms of lipid metabolism to exercising before breakfast. So as I said, I think there's a lot of work still to be done. I think it could be potentially for certain populations, potentially, especially those with disrupted sleeping patterns or severely disrupted circadian rhythms. They might benefit from exercise timing kind of optimization in particular. I think for the general public, the time you exercise doesn't really matter, because exercise is so good for you, it's so beneficial that you just try and do it whenever you can and whenever best fits into your day. If you want to be absolutely optimal, and it probably won't make much difference, but if you want to absolutely optimize your training for health, then try and do a mix of different times. Sometimes in the morning, sometimes in the evening. I think is probably what the sort of best advice we can give from the literature so far. But I think it doesn't really make that much difference. Just try and exercise whenever you can, basically. So yeah, I think that's all we can conclude from these kind of preliminary studies so far.
Yeah, no, that's a good conclusion. So what kind of studies you have in mind for the future? What kind of studies you have planned with your group?
So I think so. One study we're working on at the moment, we've actually just sort of finalized that into a manuscript, but there'll still be work to be done on that. Is really looking at this molecular effect in diabetic muscle. So again, we see this diabetic muscle that has this altered sort of downregulated time keeping ability, basically. So they can't keep time as well. That seems to also relate to their mitochondrial metabolism. So the mitochondrial metabolism oscillates in vivo and in the body and in the cells as well, and when we take the cells out of the body. And that ability again is downregulated in diabetics. And when we look at the sort of interaction, there seems to be controlled by these core clock genes directly right into the inner membrane of the mitochondria. So right into the core, this sort of powerhouse of the mitochondria, where they're sort of breaking down substrates, and they're really producing this kind of chemiosmotic, so this almost electrical current, which generates this process by which ATP is made. And the clock genes seem to be directly talking to that, and vice versa. When we manipulate mitochondrial genes within that control that inner mitochondrial membrane, we see that regulates molecules that the mitochondria produce, which then change the regulation of genes within the nucleus. So there's this sort of crosstalk directly from the nucleus of the cell to the mitochondria, and then back from the mitochondria to the nucleus. And that process seems to be disrupted in diabetics, so that they don't maintain this balance, this tuning between the clock within the nucleus and the mitochondrial sort of rhythms as well. So that could have consequences in terms of, we often see it in vivo studies showing that diabetics have reduced metabolic flexibility. So what that means is they don't have the ability to burn through sort of metabolites and substrates after a meal. And this may contribute to that, so that, they're not having these clock regulation in the morning, which is turning on the necessary pathways in order to deal with breakfast, with lunch, etc. And potentially then you're getting, impaired storage of some of those substrates and stuff. So that could contribute as well. And then we also we would like to follow up and tie down a little bit more the mechanisms by which our diabetic subjects were having this increase in glycemia after morning exercise, and really tie down why that was exactly. So as I said, we're not really sure, but we'd like to do, more detailed, controlled kind of metabolic studies to really look at exactly what's going on, whether that's something specific to diabetics, whether that's just a response that they need to be acclimated for, so something that, that they would adapt to eventually, or whether this is something we actually, need to know for exercise recommendations in diabetics.
Yeah, so very interesting studies coming in the future. I think it was really pleasure talking with you and learning about cell clocks and their relation to metabolism. So thank you for taking the time to be guest in this podcast. It was really interesting.
Thanks, Ollie. It was my pleasure. Thanks for inviting me on.
This podcast is sponsored by Fibion. Get scientific validation and learn more about Fibion at fibion.com/research. The Physical Activity Researcher podcast has created an activity tracker purchase guide for researchers. Get your free copy from the link in the podcast description.
Thank you for listening to the Physical Activity Researcher podcast.